Guten Abend, hallo, heute zum 9. Vortrag der TU for Future. Und heute ist auch der letzte Vortrag, bevor wir dann in eine Weihnachts-Winterpause gehen und uns dann erst im nächsten Jahr am 8. Januar wiedersehen. Und heute haben wir Dr. Marta Pérez Rodríguez zu Gast. Sie ist Postdoktorandin an der Abteilung Umweltgeochemie am Institut für Geoökologie. Promoviert hat sie an der Universität in Santiago de Compostela in Spanien. Ihre Forschung konzentriert sich auf den, sehen wir auch schon hinter uns, den Quecksilberkreislauf in tropischen Wäldern und die Auswirkungen der Ozeanproduktivität auf die Quecksilberdynamik. Ist sehr komplex. Der heutige Vortrag ist auf Englisch. Fragen können wie immer danach gestellt werden. Unsere Referentin heute bevorzugt allerdings die Fragen auf Englisch, aber das kriegen wir zusammen hin. I would also like to introduce Dr. Marta Pérez Rodríguez again to the internationals in the group. She is a postdoctoral researcher in the Department of Environmental Geochemistry at the Institute of Geoecology at TU Braunschweig. She completed her doctorate at the University of Santiago de Compostela in Spanien. Her research focuses on the Mercury cycle in tropical forests and the effect of ocean productivity on mercury dynamics. Today's presentation is in English. Questions can be asked afterwards, but our speaker prefers questions in English. And now I wish you an inspiring lecture und jetzt einen inspirierenden Vortrag.
Yeah, thank you very much for the introduction. So today is the last talk of the semester, and it's also maybe a little bit different for some of you. When we talk about climate change in general, we talk about how changes in precipitations, temperature will affect, for example, plant productivity, crops, how will affect different societies. Today we are going to talk about how these same effects are going to affect a pollutant. In this case, we are going to talk about a natural pollutant, that is mercury. And for that, we are going to go through different parts of the cycle, focusing in the ocean and in the tropics. So the agenda of today, we will start first with, in the first 4 points, basic knowledge about mercury as a pollutant, also as a chemical element that will help us to then follow the rest of the presentation. We will go briefly about the geochemical cycle in general, and then to introduce the topic legacy mercury. The last part and the main part of the presentation is divided into sections. The first one is from the tropics. I will talk about the role of tropical forests in general, but focus on tropical forest, on the mercury cycle, to then jump which kind of impacts climate change can have on this part of the cycle. At the end, I will go to the poles and talk about how primary productivity in the ocean, and specifically in the poles, control mercury cycling, also methyl mercury cycle, and how climate change might impact this in the future. Just a small comment, the presentation that is online contains a couple of slides more that I have removed from the presentation here just to make a better flow. Please write questions if something is not clear. So mercury as a global pollutant. So mercury is a natural element with a symbol Hg, Quecksilber in German, that we can find in different parts of our different ecosystem. It's important to note that it's the only metallic element that is liquid at a standard temperature and pressure, and this means that at room temperature, exposed mercury can volatilize. So in this room right now, there is a small amount of mercury in the gas form. When we talk about mercury in gas form, we are going to denote with Hg0. Natural deposits of mercury occurs mainly as cinnabar, mercury sulfide, that looks like this reddish stone here, if we want to say mineral. Mercury 0 is not the only form that is relevant today for us. We are going to talk about Mercury 2. Mercury 2 is formed by the oxidation of Mercury 0. A big important characteristic of the Mercury 2 is that it's a more reactive form that is going to interact with organic matter. We will see what happened with that later. So Mercury has been used since ancient times, for example in medicine as an antifungi, but the main use of mercury has been to use for gold extraction. Mercury mixed with sediment that contains gold will form an amalgam. This means like a kind of mixture between both metals. And once we burn this mass, this mixture of sediment, mercury and gold, we can evaporate the mercury because it's a metal that can evaporate, and then we will find the small pieces of gold. This is a method that is used since Roman times, but is still used today. You can see here two pictures of what is called artisanal and small-scale gold mining that is take place mainly Latin America and Central Africa. The big problem of using mercury in this way is that mercury is relatively cheap, so they are not so much concerned about recuperating the mercury they are using, and a big part of the mercury is lost to the atmosphere. So we said that artisanal and small-scale gold mining is a big challenge in terms of mercury pollution, even nowadays. Another form of mercury we are going to talk today is methyl mercury. This is an organic form of mercury. It comes from the methylation, that is the bind of a methyl group, carbon with 3 H forms to a mercury form. This is done by bacteria in anaerobic conditions, for example, sediments. Anaerobic conditions means in conditions where the oxygen is limited. Mercury, and especially methyl mercury, are neurotoxins. They are going to affect our neural system, our kidneys also, and will affect the development of babies when they are still in the body of the mom. This is one of the reasons why it's not recommended for pregnant women to eat tuna or these big fish predators during the pregnancy. Here I add some information. You can check yourself about what is the Minamata disease. Minamata disease was a disease found in the area of Minamata, Japan, after a company was depositing during several years mercury as part of wastewater to the bay, to the ocean, and there in the sediments methyl mercury was formed. After some years it started to appear several cases in the local population, in babies with problems with development, but also in adults and old people. The reason was this population was eating fish and sea fish from the bay as a local source of food, and these fish and seafood, they were contaminated with methyl mercury. The Minamata Convention is an agreement that tries to protect human health and environment from anthropogenic emissions and releases of mercury and mercury compounds. It's signed for more than 115 countries and is the base of mercury protection. Okay, when we talk about the geochemical cycle of mercury, I want to distinguish several terms that I will be using along the presentation. The first ones are what are sources. Obviously, sources are all of those processes that are going to release mercury from the atmosphere, the hydrosphere, biosphere, out to the atmosphere, to the environment. We can distinguish between natural and anthropogenic, don't need definition, and between primary and secondary. So the main natural source of mercury are volcanoes, rock weathering, and geothermal activity. During this process, mercury in different forms is released to the atmosphere. We also can find biomass burning, forest biomass burning, when this fire is natural, but of course can also be anthropogenic. Main anthropogenic emissions are related with mining activities, but also with the combustion of fossil fuels and some smelting activities. And there will be other evasion processes that we will see slightly during the presentation. What might not be so obvious is what are sinks. So sinks are going to be all of those compartments that in a temporal or permanent way are going to store mercury. Temporal ones are the forest biomass, as we mentioned, that we mentioned already here, and the surface soils. So this means the first centimeters of soil. As a permanent are marine sediments and deep soils. What means permanence? Means that once the mercury reaches this compartment, it's going to be stored there for a long period of time, almost, we can say, permanently. This is how it looks, the mercury cycle, and maybe you already got the idea that mercury is an element that somehow jumps from one compartment to the other. So we can get, for example, emissions of mercury due to volcanic activity to the atmosphere. That is the main way to transport mercury from one place to the other. Elemental mercury, for example, can be up to 1 year in the atmosphere. Some for different process is going to be deposited and can be emitted again, for example, as we mentioned before, by a biomass burning. Ocean is a big, represent a big part of the mercury cycle, however, is not very well understood. Due to this process of emissions and depositions, we have been realized that the mercury that we are emitting now is going to continue in this cycle for several years. And then is when we talk about legacy mercury. So the mercury that we are seeing now in our environments is the product of past activities. For example, right now we are starting to see the decrease of mercury that was started in the 70s. So there is a big gap between emissions and finally somehow cycling. How long is the element in cycle? So when we stop in the 70s emitting mercury, or we reduce due to different policies, we are seeing now the effects. Think about that. I don't want to enter so much in the big numbers of mercury fluxes. I just wanted to point you that you see more or less how high are the natural primary emissions, for example, just from the, for example, volcanic activity, weathering, and so on, that we have 200 megagrams of mercury per year, compared to, for example, anthropogenic activities. This is a relatively recent paper, and this value has already been decreased in the last 30 years. And as I mentioned before, how incredible are the fluxes out and into the ocean. So with this more or less understand, we are going to start in the tropics. So the first thing that we need to understand is what is the relation between vegetation and mercury. Maybe some of you recognize what is this, maybe others not. This is an stomata. So what is an stomata? It's a small pore, usually found in the leaves of vegetation, but also can be in other parts of vegetation, that is in charge, or is where occurs the transfer of gases. So this means that these pores, when are open, CO2 can enter into the plant to take place the photosynthesis, and oxygen is released. The opening and closing of the stomata or stoma is controlled by these guard cells that, in a very simple explanation, is going to depend, I hope there is no plant physiology here, but is going to be work based on what is the water stress of the plant. So if the plant is stressed, unless water is available, these guard cells are going to close, because at the same time that it's taking CO2, also oxygen, also water can be lost. So it's a mechanism of protection. When the conditions are fine, stomata are open. Of course, there is this day-night process, but in normal conditions, they are open and gas transfer can take place. At the same time that we have CO2 entering in through the stomata, we are also going to have elemental mercury. So our gas that is in the atmosphere is going to enter passively through the stomata. The plant doesn't need the mercury, the plant doesn't want the mercury, but it just enters. Not all the mercury that is entering through the stomata is staying in the plant. So here we have a little bit a more complicated scheme about how works the mercury uptake in plants. So here we have a stomatal uptake, or stomal uptake of mercury 0, and 30% of this uptake can be lost. So can enter, and if it is not stabilized, comes out again. Part of this mercury is going to be transformed in mercury 2, like we saw before, and once it transferred to mercury 2, it's more or less stable there and will accumulate in leaves. Apart from this stomatal uptake or stomal uptake, we are going to have also some process that occurs on the surface of the leaf. So there is a small amount of mercury that also can get attached on the surface. Part is going to be remitted, because as we said before, mercury 0 is very unstable. So when temperature is nice, when there is enough light, it's going to be re-emitted to the atmosphere. And then I also want to point that this atmospheric uptake constitutes 89% of the mercury that is taken by plants, but we still have a small root uptake that we will not talk about this today, but is there. What processes are going to affect how much mercury is entering into the leaf? Because it's a passive process, one of the main factors are going to be atmospheric concentrations. So when we have higher atmospheric concentrations, the amount of mercury that easily will enter into the leaf is going to be higher. There are other parameters, like for example, the temperature, the number of stomata, and the leaf age, and then a lot of different leaf physiological parameters. So for example, stomatal conduction, rate of photosynthesis, and so on. Very nice here, this description, but there are few papers just describing what can happen. So this means that it's actually not 100% clear how each of these parameters affect the amount of mercury that is taken by the leaf. However, one that is clearly demonstrate and assumes are atmospheric concentration and leaf age. For example, in this case, it's a very easy and nice example. So we have in the horizontal axis the leaf age in days from 0 to 50, and in the vertical axis mercury concentrations, foliar mercury concentrations. In red, we have a plant growing in high atmospheric concentration, and in blue, a plant growing in lower atmospheric mercury concentrations. So there is already this mismatch, this separation between the two groups, and in both cases, the increase of the leaf age increase mercury concentration. And this is logic. When we have something that enters passively and accumulates there and is not going to release, as older as is the leaf, as more mercury is going to pass through this process. And now the question is, how important is the role of vegetation of the global mercury cycle? So we see that mercury can interact with vegetation, but in a global scale, how relevant is that? And here we go through three different locations: Norway, Germany, and Finland. And in the three cases, they are measuring CO2 concentrations in the atmosphere, mercury zero concentrations also in the atmosphere, and a long time, around between 2010 and 2016 or 14. And what we can see now here is a very nice covariance between both parameters. So this means that in the periods where we have high CO2 concentrations, we also have high mercury concentrations, and in the other way around. So this means the process, like vegetation, that can modulate seasonally the amount of CO2, that is something already known, also regulate the amount of mercury in the atmosphere just by uptake. This was also observed in the same kind of study in Canada, but not in South Africa or Amsterdam Island, that belongs to France, but it's a small island in the middle of the Indic Ocean. What is the reason? In these three locations, we have a very strong seasonal impact of vegetation. We are in temperate boreal ecosystems. In this case, the amount of vegetation is really limited and is also an island surrounded by ocean all around, so other processes are more relevant. And in the case of South Africa, in general also the own CO2 variation seasonally is not so strong, so doesn't work so well. A small sum up, so we don't lose our points. So up to now we have seen how mercury gas is taken up by plants, mainly through stomata and from the atmosphere. This uptake is, or how much is on leaf, is related with atmospheric concentrations and others, including plant physiological process, environmental conditions. Mercury is going to be stored in leaves, and this uptake has a strong global seasonal effect, or global effect in areas with high seasonality. And then comes again the question, why is this important? What does this mean for forest ecosystems and the mercury cycle in general, and in tropical areas, and climate change? So just only one small piece more is that the mercury that is stored in leaves is not going to be there forever. At some point, this tree will lose the leaves, and this leaf will go to the soil. So this process, like through the litter fall, so this process of transporting mercury from the atmosphere to the canopy, and then with the leaf to the soil, brings around 1,100 megagrams of mercury per year to the terrestrial ecosystems. New research claims these values are higher. And this is a very high number when we consider that emissions from anthropogenic sources are around the 2,000 megagrams per year. So a big part of how much is emitted anthropogenically every year is taken up by forest. And 70% of this deposition occurs in tropical and subtropical regions. The reason is tropical regions have amazing weather. They have sun the whole year around. In many areas they have enough precipitation. So conditions for growing vegetation is amazing. So they produce way more biomass than the one we have in temperate areas. And just because of that, this number is so high. This is a very nice map from a publication of 2017. There has been some updates, but there is not nicer map than this one that shows mercury deposition through litter fall worldwide, and you can see how tropical and subtropical areas are just glowing. How high are the differences between 70 and 200 found, for example, in the Amazon, Congo, or in East Asia, compared to, for example, what we have in temperate areas. When we check which kind of forests are those, we can see that this big yellow percent are the tropical and subtropical moist broadleaf forests. So these are more or less like typical rainforest, Amazonia. Here in green, this smaller portion here are tropical and subtropical grasslands and savanna. So you can think about a typical image of a savanna in Africa, but also includes other forests that, for example, suffers a period of dry season and rain season. And in blue, I just marked temperate and broadleaf mixed forest. For example, the ones that we can find in Europe, North America, and so on. So we are going to have like a high mercury deposition through litterfall, but it's not all, because this litterfall at the end is in contact with the soil. Here I make this somehow easy, understandable, hopefully, a scheme of what are the processes that will affect once the leaves are deposited. So we mentioned that there is going to be deposition of leaf with mercury. The leaves that are going to be here, they will suffer processes of decomposition, the degradation of organic matter, and this will re-emit some mercury that will get free. Other part of the mercury that is somehow mobilized during this degradation of organic matter will hopefully, a small part, go to the deep soil, and we can consider then that is a final sink. So we'll get stored and bound there for a longer period of time. But then, another part will get lost through rivers and get as a dissolved mercury to rivers and the ocean. So all of these processes here are going to control how much we get stored, how much goes away in the form of dissolved, and how much is re-emitted. This is why tropical forests, or forests in general, are considered temporal sinks. So during a certain period of time, they retain the mercury there, but immediately different parts are going to be re-emitted. In this figure here represents the amount of mercury stored in surface soils. Before we have litterfall, and here surface soils, the upper 30 centimeters. And just with a quick look, we can see that we have a similar pattern than we have before. So tropical and subtropical areas where litterfall deposition, mercury litterfall deposition was high, they are going to also have high mercury accumulations in the top soil. And now comes the effect of climate change, how climate change will affect all of these processes in tropical forest. So the first thing I want to say is that there is a big uncertainty. So the study of tropical forest, has started, I can say, maybe 10 years ago. So we have a bunch of literature about temporal, boreal soils. We have even studies about remote areas in southern Patagonia, everywhere. Tropical forests have received little attention up to now. The reason, of course, is easier to go here, drive 100 kilometers and pick up some samples instead of going to the Amazonia, East Asia, or Congo Basin to do research. The main effects that are expected to happen in tropical regions are relatively changes in temperature, mainly changes in precipitation patterns, so this means big difference between dry and wet season, and variations in El Niño southern oscillations. Apart from climate change, I wanted to mention also other environmental changes directly and immediately provoked by human activity, like can be deforestation and forest fires. So I put both together just to see what can be the final effect. So as we saw before, Amazon is currently important net sink for mercury. So net sink means that between this uptake and remission, there is one part that stays there in the soils. And one of the big concerns that can happen due to climate change is the transition between a rainforest, like more or less we have this picture here, to a savanna. That will be something similar like this. So we will not expect elephants in the Amazonas, even if transformed into a savanna, but it will look something like, for example, this Cerrado ecosystem that is already on the limits of the Amazonia in Brazil. And there are two tipping points, so 2 points that can provoke that there is a complete transition between these kind of forest types. First, that there is an increase of 4 degrees above pre-industrial temperatures, and both options, if there is a more than between 20 and 40% deforestation in the region. The reasons are that if any of both happens, the regional moisture recycling gets weak and is not enough water available to maintain a rainforest like we know. And what will happen with mercury? So here we have a publication, or result from a publication for 2022. So also quite new, modeling what will happen in those conditions. So we have here rainforest transforming into savanna. And what we see here is mercury zero dry deposition velocity. That is more or less an indication of how much is deposited there. Then we see that right now, we have depositions between 0.3 or 0.25 to 0.4, and this deposition, so this uptake of mercury almost disappear with when the rainforest transform into a savanna ecosystem. And the question is, where is going all of this mercury? So according to this model, an additional 280 megagrams of mercury per year will be deposited into the closest ocean. So everything that is not deposited on one side at the end will be deposited somewhere else. In this case, the ocean. Apart from this transformation between savanna and rainforest to savanna, we have also the direct problem of deforestation. So if deforestation continues as it is now, the emissions from the region will increase by 153 megagrams per year by 2015, and again, this mercury will deposit somewhere else, in this case, the ocean. In this small map, you can see the emissions due to mercury emissions due to deforestation worldwide, and how the different countries were contributing to them. So we can summarize and take some small take-home message to bring up this somehow together. So first, we know, and it's clear, that forests are a major sink of mercury deposition to the terrestrial ecosystem, mainly through stomatal uptake and litterfall. Mercury accumulates in the soils, and this accumulation is going to be strongly connected with mercury uptake, although some losses are going to occur directly through the atmosphere or in a dissolved phase through the rivers and oceans. Tropical forests are significant due to the high biomass and litterfall production, and climate change and deforestation may reduce the tropical forest's ability to absorb this atmospheric mercury, increasing ocean deposition. And just a last paper I found preparing this presentation, it is expected that, like global forests, so in general, this, due to climate change, there will be a reduction of 60% of mercury uptake from the atmosphere, and this mercury will go to the ocean. Apart from the differences due to litter fall and changes in the forest type, increasing temperatures may also destabilize surface soil pools, increasing the emissions to the atmosphere, but also through the rivers and the oceans. Now, I didn't want to miss the opportunity to talk us a little bit about what are we doing here at the TU Braunschweig, and talk just briefly about our Forest Mercury project. So what we're doing here is we are trying to go a little bit more into detail what are the differences between different tropical forests in terms of mercury uptake, mercury accumulations in the biomass, transferred to the litter fall and soils. Because, as I mentioned before, even if we have all of these very nice global maps about mercury in different parts, when you go to the raw data and you see how many locations actually they have in tropical areas, is less than 2-3% of the total collection. So our point here is to see what happens when we have a tropical dry forest, so where we have some seasonality, in comparison to a tropical rainforest that means no seasonality. We are studying different areas in both forests and collecting all the different parts that can help us to understand how much mercury are actually these forests taking up. So we are collecting leaves, soils, litter fall, but also measuring, for example, how much mercury is in the atmosphere. Right now, we are done with the second sampling campaign, and May next year we will be done with the last and third. Maybe sometime we can talk a little bit more about the results from the project. So, this was our travel from the tropics, and now we will go to the poles and to the ocean. We already mentioned before that what is not uptake by the vegetation, by the forest, goes to the ocean. So atmospheric deposition on the oceans are high compared to the terrestrial ecosystem. I don't know if you remember before that we have around 1,000 something megagrams per year. We are talking about here 6,000 megagrams, and a net burial, so this means somehow like a net uptake and stay there of around 1,000. If you compare these numbers with the one I am saying, and you find in different mercury cycles, the values are more or less. So these estimations are generally really raw, but give us an idea of how important are different parts. Oceans are also important because the main vector for main way of methyl mercury exposure in humans, here in the US in these publications, but in general, is through fish and shellfish consumption. And 60% of them comes from the ocean. And then another important point is that ocean sediments are considered one of the final mercury sinks. I put this picture here because in this case, one of the most important parts of what's happening in the ocean is going to happens in the upper part, in what is called the photic zone. Photic zone is this upper part layer of a body water, can be also a lake, but we are talking here about ocean, that is the one that receives the sunlight, is the one that allows phytoplankton. This means this is small microalgae that produce most of the do most of the photosynthesis in the ocean and are the main source of primary productivity. And everything happens here, but what happens here in this upper 100 to 200 meters have an effect not only in the ocean but also in the terrestrial ecosystems. As I mentioned before, we have in this upper part phytoplankton that are responsible for the majority of the primary production, but they are also, and because of that, they are the base of the marine food web. So they are the entrance, the beginning. So they are the ones that are going to be eaten by zooplankton, this by another one, and then increasing until we eat a fish if we eat fish. Sunlight, CO2, and nutrients will affect this primary production. Here in this cute picture, we have Antarctic diatoms that are a type of phytoplankton that is encapsulated or have like a silicon structure. And we are going to see them that are also relevant in terms of mercury accumulation. But primary productivity is not constant in the ocean. So we have in this map here chlorophyll concentration, so this pigment that indicates like the capacity of doing photosynthesis. And the good thing with chlorophyll is that can be observed with satellite. So this is a satellite composite image. And we can distinguish here different parts of the ocean. So first, you can see on the west of the continents, for example, here in the west of Africa, the west of South America, areas with high productivity. These are upwelling areas, are areas with the deep, rich in nutrients, cold waters, come from the bottom of the ocean and go up. This is due to currents, surface winds, and so on. What will produce high primary productivity? Because the conditions are good for that. And what are related to most of these areas are fisheries areas. So are areas where fish is abundant, and we go there and fish them. Then we have polar areas. They are going to be also rich in nutrients and with a strong seasonality. We can see here the Arctic area, also quite green, and also in the Southern Ocean, Antarctic area, also quite green. And then we are going to find the ocean deserts. Are all of these blue areas here that are most of them in the subtropical, tropical areas. And in these parts, the conditions, for example, water stratification, doesn't allow a very good nutrient cycle, so are relatively poor in nutrients. So productivity is also small. Some of the first ideas or main points that brought to study mercury in the marine ocean in more detail started with the Minamata accident we mentioned before, and in general, high mercury concentrations are found in fishes everywhere, and also as soon as we increase the trophic level in other animals. The reason is that mercury, and methyl mercury in particular, is bioaccumulates. So this means that it cannot be removed from the body once it's uptake, and then as soon as we increase the trophic level, this amount increases. The traditional approach, that is what one tries to represent this very simple figure, is that the methyl mercury that we found in fish and other animals, marine animals, was coming from the methylation of mercury into the sediments. However, the average depth of the ocean is more than 3,000 meters. And we have several areas, 4,000, 5,000. And then start to come the question, is this really likely that we are having methyl mercury coming back from there, from 4,000 meters, going to the surface and be in contact with fishes? So 4 kilometers of water were separating the source of methyl mercury to the organisms taking them. So this that somehow was accepted in lakes was discarded relatively soon in the ocean. So then the next and current approach was, okay, this is not likely. Something might be happening here in the upper ocean where productivity takes place that produce the methyl mercury somehow that at some point will enter in the trophic web. And now we need to step out for some a minute from the ocean and go to lakes. The reason is that research in oceans has been developing very, very slowly. And I can say in terms of Mercury, in the last 8 years, because it's extremely expensive to go there. You require like a ship, you require a lot of people, a lot of money, and concentrations of everything are very low. So some of the first ideas that have appeared or have been developed in the ocean in the last 10 years come always from the studies in lakes. They are easier to study, it's cheaper, and concentrations are in general higher, easy to measure. So the first thing that came from lakes, or one of the things that came from lakes, a part of this methyl mercury from the sediment, is what was called the algae scavenging hypothesis. This hypothesis says that mercury that is in the water column and enters in contact with our algae production in the surface is bring down, so it's a scavenge, so it's collected from the water column and bring down to the sediments. This was observed, for example, in an Arctic lake in 2000. It's a study from 2008, where you can see here in the vertical axis, this is time, because as you might know, when you collect a sediment in a lake, you can use as an archive, because we know what this is on top has been more recently deposited, and as soon as you go deeper, it's older and older. With the right techniques, you can get an idea how old are you going down. In blue, we have S2 carbon, that in this case is an indicator of an organic matter related with algae and productivity, and then we have here total carbon concentrations that are in this axis. And what they observe is that increases in S2 carbon, so in the algae organic matter, correlate with the amount of mercury. So when we have higher algae, higher total mercury concentration. And then the question was, what is happening in the ocean? Is this happening, and how important is that? Here, just I want to show you in this figure, we have different kind of primary productors, so different phytoplankton types by color, that you can see that they somehow dominate one area. And I want that you check about diatoms that are this reddish color here on the bottom in Antarctica and also in the Arctic, are the ones we mentioned before. Here there is a big ring around Antarctica full of sediments containing mainly diatom remain. Is what is called the diatomous belt, and it's a huge surface. You need to think that Antarctica is 40% bigger than Europe. There is this interesting figure showing more or less the effect, and this ring is even around Antarctica. So we are having a huge ring of deposits full of diatoms. So in 2018, as a part of the PhD of Sara Zaferani, here at the University of Braunschweig, we analyzed 3 cores from 3 different locations in Antarctica, and we measure mercury concentrations. Our idea was to check how much mercury is there. Can we estimate how much mercury is accumulated in this diatomous belt? So we use sedimentation rates. This means how much sediment is accumulated per year, mercury concentrations, and 2 other independent approaches that I will not go through them, to estimate how much mercury was in these sediments. So our results show that mercury, based on this calculation, the mercury accumulation just in this part of the ocean was 7 times higher than previous estimates. So previous estimates were, or there was a mismatch with emissions and accumulations, or somehow this mechanism was not considered before. And then based on these estimations and emission sources, 20% of the anthropogenic emissions, global, might be accumulated just in this area of Antarctica. Apart from these estimations, we also look at, again, temporally change. So this is one of the cores of this study, and here again we have years. So we know that the surface goes in the upper part, and as soon as you go deep into the sediment, you also can get like older sections, and with the right method getting an age. What we found here also that is in Antarctica, there was the first signs of mercury pollution around 150 years ago, so with the beginning of the industrialization. So at the same time, more or less, that we started to make global pollution here in Europe with industrialization and North America, this mercury was reaching Antarctica. And this indicates a fast mechanism. So a fast mechanism was able to first, of course, transport through the atmosphere of the mercury zero, but also in a fast way a catch from the atmosphere to the water column and then to the sediment. In a subsequent study, Sara Zafferani proposed a mechanism of fast sinking diatom particles, removing dissolved water phase mercury from the water column through the scavenging. So what we are seeing here is this algae scavenging hypothesis observed in lake was also shown to happen in our diatom ooze sediments. One of the studies afterwards showed that these small variations that we observe also around the record might be related with the melting of the ice. So in period where the ice was covering the whole bay of Adelie Bay, that is this core, no algae was formed, so no mercury was taken up to the sediment. In periods where this ice was broken, conditions allowed that there was a good flux of mercury from the atmosphere to the water, and this algae to be able to produce because enough sunlight and fast mechanism bringing them to the sediment. So somehow we could say that the scavenging takes place in the ocean under certain conditions. But now the question is again, what is happening here, how the methylmercury is formed, and how does it enter into the trophic web. I want just to point slightly like that we have been talking about methylmercury, but actually we have different methylmercury forms. So we have what is usually called as methylmercury or monomethylmercury, that is this form here, that is the one that is going to be able to interact with all the organic matter. And then we have another form, that is the one called dimethylmercury, because we have two methyl groups here, that this one is going to stay dissolved, and the transformation between both is dynamic. And we can have process of methylation and demethylation. I don't want you to forget that the mercury that we need to do this first methylation is mercury 2, that is the reactive one, and this will come from the one that is in the atmosphere. This monomethylmercury or methylmercury, the one with only one methyl, is the one that can interact with organic matter and is the one that will bioaccumulate. And then we come to what is discussed now as what is the mercury model in the ocean. Explosion. So this is a theory model that includes all these different parts I have been mentioning here. And I just want you to show you which different, how this compares to the simple model we did before, and what are the main questions right now. Because this model is not solved. So this is a theoretical model. There are some constants measured, but it's not known how does this really happens. So the first thing we have here is our Mercury 0, the one that we get from the atmosphere as a gas, but we also can get Mercury 2 that is already in the atmosphere as a dissolved phase. This will enter into the ocean. This Mercury 0 also can get as our Mercury 2 dissolved phase. Okay, this can go in both ways. So this Mercury 2 can transform to 0 and so on. And then now we have here a big and important step. Mercury that is dissolved in the water can also can interact also with particulate matter. This particulate matter can be, for example, phytoplankton, but can also be fecal pellets, can be also every kind of particulate organic matter that is in the water column. And in these conditions, when we have this Mercury 2 in a particulate phase, so in contact with the organic matter, is going to create the perfect environment for methylation. So methylation will take place at some point somewhere here. When, we still don't know. First indications show that this happens when this particulate gets decomposed. Why this will happen? We need, for methylation to take place, as we mentioned before, we need some bacteria. And these bacteria to live and to produce reaction, they need food. The food they can find in the ocean are mainly in small particles, or particles in general. But not all the methyl mercury that is in the particles will stay and sink, like mercury we have here. If not, that during this degradation, some mercury will get dissolved, so we will have also methyl mercury dissolved, and this dimethyl mercury also dissolve. The process on which conditions all of this interaction happens is still part of the discussion, and in the last 2-3 years have been changed so much that this paper from 2022, what is already written there, apart from this nice figure, is almost completely wrong. So I cannot really explain you how is this happening. I only want to point you out which are the fractions and parts that are relevant. But what we really measure, so as I said, we don't know what is happening, we don't know in which conditions it's happening, but we are measuring things that help us to try to understand what is behind. So for example, these are typical profiles. In this case, they are slightly modeled to show like a nicer curve of, for example, methyl mercury concentrations in the water column. Some things that you can consider, for example, is that here we have from the surface to the 600 meters, and here in different periods of the month of the year. And then you can see, for example, that in winter we have like a very straight line in the surface, while in the other month there's this nice curve. This is related of how the strong winds in the winter mix all the surface of the ocean, limiting the formation, for example, of different layers. Another thing that we can observe, for example, in this one, is that how in a conditions where there is low primary productivity and low nutrient, maximum of methyl mercury concentrations are significantly lower than, for example, in this case where we have higher mercury concentrations. The current hypothesis are that these trends here in the upper layer, so this somehow lower minimum in the surface and increasing, are related of where methyl mercury is produced and where is released. On the other hand, we have not only the dissolved part we mentioned before, but also the particulate part. Just only two considerations here. The first one is that I don't know how many of you are familiar with chemical units, but these are measured, all of these values are measured in picomole of methyl mercury. This amount between 0.1 to 0.3 is nothing. So it's nothing. It's so difficult to measure that this is possible since 8 years ago, more or less, that the techniques were developed enough to be able to measure these levels. Everything what is measured before can also more or less consider as contaminated. And the same in the case of the particulate matter. Values are extremely low. However, despite the values are low in the particulates, despite the values are low in the water phase, these are values of methyl mercury on the right side, total mercury on the left side, in invertebrates and fish, so invertebrates on the top, fish on the bottom, that you see how with the increase of the trophic level, that are these horizontal axes, increase slightly as soon as the animal starts, or we have like a higher trophic level. Just only a small comment, something that brings a little bit more complexity of this cycle. And in this figure, I only want to point this thing. Up to now, we have considered like mainly the part of the diatoms, and then we consider zooplankton and also other fishes just as an end part of the system. However, zooplankton constitutes one of the biggest migrations mechanisms of nutrients because they live in the dark place and they migrate every day 600 meters to feed, and then they die here, but they also release leftovers after eating, and this constitutes a big source of nutrients, but also mercury to the system. So, with all of this big uncertainty, what will happen with climate change is even more uncertain. So, climate change might change water temperature, change wind intensity, will have effects on productivity up to date and in a long term, changes in nutrient cycle and ice cover. I want to just to point you here how uncertain are all of our results in general of what will be the effect of climate change. So for example, two very simple variables, changes in temperature, changes in wind speed. How will they affect mercury concentrations? So just only changes in temperature. When we have high warmer water, they can absorb, they can contain less gas, less mercury. Wind speed, when we have no wind, the mix is worse, so the sea exchange reduces, so there will be less losses. And then we have two completely different results of how climate change will affect the ocean, just changing two relatively easy, understandable variables. So based on changes in temperature, by 2100, the surface ocean will contain less mercury. By changes in wind speed, most of the ocean, with some exemptions, will have high or significantly high more mercury. The same will happen with changes in wind intensity and effects on productivity. When we'll not have like this wind mixing, this cycling of nutrients is going to get lower and productivity will not take place. So this means that some of these patterns will change. How been changed up to now is that we have enough nutrients, but a long-term perspective says that in general, with the exception of some regions in the Arctic, most of the ocean will decrease the productivity. Another example is how climate change might affect is, for example, the effect of the loss of the ice. So up to now, the loss of the ice is relatively seasonal. So we have seasons with a little bit less ice, but the ice next year comes back again most of the time. So in these conditions, productivity still takes place and is somehow and has rise when the ice retreats. Long-term perspective says that once all the ice is gone, at some point nutrient cycling will decrease, disappear, and then instead of having more scavenging, like we will have in normal conditions, more mercury can get evaporated and will increase the evasion. So we come back to a small take-home message. Difficult to do because the state of the research is relatively on the beginning and there are many current contradictions, but for sure we can say that oceans are main sink of temporal sink of atmospheric deposition. Around 20% or a big percent of the anthropogenic emission is stored in Antarctica diatomous belts or in all those areas with high productivity. Ocean productivity influence mercury cycling by scavenging from the surface of the mercury, creating conditions for methyl mercury, but also be the first step of the marine web for methyl mercury and mercury accumulations. We know, or is likely, that climate change will impact all of the mercury and methyl mercury cycle, but it's difficult to really say right now in which way and in which direction, and what will happen in the next 20 years and what will happen from until 2100, for example, in 100 years more. One thing that we can say for sure is that polar regions, specifically the Arctic, because of the high nutrient inputs they have through rivers, is going to be a big challenge and is going to be different from many other parts of the ocean. They have unique conditions. And then, like before, I don't want to finish without talking a little bit about the research we are doing here at the TU Braunschweig. We have a project to investigate primary production and the effect on the mercury cycle in the Southern Ocean. You can find these two links from the TU magazine, more information. And this brought us to the Southern Ocean in 2022, and right now we are somehow getting ready with all the results, and hopefully in some month we can say a little bit more about what we really found. What we did there is to try to make it clear again all of the different parts of this puzzle. So we have collected water samples to understand the dissolved phase. We have collected particles, mainly composed by phytoplankton, to see what is really on the particles, different kind of zooplankton to see how methyl mercury bioaccumulates, and sediments to calculate how much is falls down into the sediments and stored there. Due to this opportunity and together with the Hadel group, the Danish Center for Hadel Research, we have been also now involved in a project in the Arctic, the Fram Strait. You can find here more information, where we are studying sediments and mercury accumulation in different parts, not only in the Arctic, but also in other parts of the ocean, of the deepest part of the ocean. Just, I want to finish. Thank you first for all of you to be here, for the organization. It was a cool opportunity. I cannot not say thank you to Harald Bister. Wow. Making some effect. Yeah, thank you to his trust and also giving me so many opportunities to work here at the TU Braunschweig. And yeah, and also in all of this project. Juan Morales is mostly in charge now of the project in Costa Rica, so he's also doing a very good job. And all the team, students involved in the different projects, and the people from the cruises, especially Ronnie Glut and the team. Just two things before I finish. We are, as we said before, we are part of the Institute of Geoecology. We have our areas in our offices in Langerkamp. Here you can find us online, of course, through the university page too. And just a little bit of publicity. We have a new Instagram account. We are slowly updating and uploading new content about all of these fieldworks, the projects, not only my projects, but also the Forest Lab, a new project. So if you're interested, just please check it out, and slowly you will get more and more information. So thank you very much, all of you. And yeah, I will be happy to answer some questions if you have questions. Thank you.
Thank you so much for this very informative and alarming presentation. We have now time for some questions. Are there any questions? Be brave.
Yeah, thanks very much, Marta. I am convinced that there's a risk that we lose rainforests, and you said that this is the main thing for terrestrial uptake. But isn't it at least partly compensated by the fact that we will have an increase in forest cover in the north?
That's a tough question. I mean, I will need to see the numbers. Right now, most of the studies, so I've not done any of these models, point that is still, tropical forests are still like the main driver. So we need to think like, first that temperate forests have this season, right? So there is part of the year where there is no uptake at all. While in tropical areas, especially in all of these rainforest areas, we have the whole year, like spring conditions, if we want to say like here. And this can be seen just only for how much mercury is accumulated in the leaves. So we have in a normal forest here, we can have up to 40, 50 micrograms per kilogram. When we go to a tropical forest, we are in 200. Reason is leaf age. As longer as the leaf can live, more mercury can accumulate, and then the amount of biomass that is produced is enormous. So what will happen if we reforest the whole temperate areas? I really don't know, but it will be difficult to compensate what the numbers said, but it's a good point.
I have another one, and I think it's more entertaining. So let's assume you figure out a way to remove the mercury from the atmosphere or from the ocean. What would you do with it?
What will you do with it? I mean, you don't need to do anything. You could store it. I mean, to have an amount of mercury that is, so mercury is very dense, so you can get a lot of mercury in a very small space. I mean, you could still use it for industrial purposes. For example, there are some instruments in the lab that they have been used mercury as an indicator. If it is well used, it shouldn't be a big problem. The questions are all the losses we have through, for example, melting of metals or other industrial processes. I want to mention something interesting that maybe not you, in general, you are very young, but maybe the old people remember that some of the way to repair the teeth some years ago, these amalgams, were made out of mercury, were covered by mercury. Since 5-8 years, there was like a law to remove all of them and put like artificial ones. And every dentist must have a way to collect, clean this amalgam of mercury and be able to somehow separate from the normal garbage. So there have been people living with amalgams of teeth, or like mercury pieces in the mouth for many years and nothing happens. So elemental mercury in general is not such a big concern if we have in a stable phase. The big problem is, of course, methyl mercury. That is a big problem, and even small amounts are very toxic. But I mean, the mercury had some uses, and well use shouldn't be a problem. The problem is that it's difficult to avoid losses of mercury or maluses or bad storage, waste, and so on. The questions can be also in German, if this helps. No problem. Then can I ask a question to the public? So now someone has to answer. So which part of the whole topic was known for you? That's my first question. And the second question is, what was the most somehow surprising part of this presentation? Egal.
Okay, dann würde ich gerne in Deutsch.
Ja.
Also überraschend war sozusagen, dass um die Antarktis besonders hohe Ablagerungen des Quecksilbers vorkommen. Und dann hätte ich noch eine andere Frage. Würden Sie denn empfehlen, sozusagen noch, abgesehen davon, dass es nicht besonders gesund ist, Seefisch zu essen, oder?
So, I mean, I come from a part of Spain where fish and sea fish is one of the somehow main dishes I have been eating all my life. In general conditions, you can eat fish and seafood without a problem if it is not from a contaminated part. The main risks are for big predators, for example, this red tuna that is used for sushi, for example, and is so common in Japan, or all of these big fishes. And especially when they are consumed during the pregnancy, so it's something that actually doctors recommend not to do. This can be a problem because due to this biomagnification, how much methyl mercury is accumulated and increased during the trophic web, the values get like in dangerous level, especially for the baby that is growing up there. For adults, eating seafood or fish in a relatively low amount is not a problem in normal conditions. I am also not a medical doctor, but don't understand me wrong. But I mean, we have seen like people eat fish and seafood, and in general, nothing happens. I can think about now, some years ago was the first time I heard someone that was intoxicated with methyl mercury just by normal fish consumption. Was someone from Spain, like I think was a presenter from TV that started to like a diet very rich in tuna, in this red tuna, as part of a training program with a lot of muscle building and so on, and consuming every day for, I don't know what was the period, couple of years, 2-3 years, only like tuna every day. And after that, I think 2-3 years, they discovered that he was intoxicated by methyl mercury. So it was a very extreme case of consumption of a big predator every single day. So the intoxication by methyl mercury in normal conditions is, for us here, is the risk is low. It's different, for example, from populations from the Arctic. For example, we need to think Inuit and other communities that they have a big part of, or an important part of their food consumption are seals, whales, and all of these big mammals. And then they also especially use parts like the liver, that is a place where the mercury is methyl mercury is accumulated. So for those populations, the consumption of fish or mammals in this case is a problem. For most of us in a normal consumption, for that part shouldn't be a problem.
Okay, so, yeah, okay.
Sorry.
No, kein Problem.
Wieder in Deutsch. Und zwar, Sie hatten ja angesprochen, dass bei den Zahnärzten und Zahnärztinnen sehr darauf geachtet wird, dass das Quecksilber abgeschieden wird. Gibt es dann irgendwelche Vorschriften, Grenzwerte und so weiter bei der Verbrennung von fossilen Brennstoffen, also beim Verbrennungsprozess von Kohle, was glaube ich auch ein großer, wie Sie sagten, also wenn man organische Substanzen verbrennt, eine große Quelle von Quecksilberausscheidung ist?
Can someone help me? I don't know if I understood right. So if the mercury removed from the teeth, sorry. Yeah, so there are regulations. Most of the mercury regulations are based on this Minamata Convention. So the main regulations are based on just because of the coal burning. But one of the reasons is because in coal you can find find mercury, and due to the burning this is lost. So I don't know if there are specific regulations checking the quality of the coal before it's burned, but I know that where the coal is burned or where it's burned, it should have filters to prevent that this mercury is released. So I don't know if they need to check like the concentrations in this coal. I don't think so, but they need to have specific filters to change these filters quite often and to prevent these emissions. I can also have a small example from where I come from. In the north of Spain, there was a thermic plant using coal for many years to produce electricity, and they were able to find the signature of the coal that was burned there, not in the north of Spain, but in Norway. So you have to think that these plants, they have very high towers, chimneys, where the gas are released. So they are somehow not in contact with the somehow local winds, if you want to say like that. So they are emitted really high in the atmosphere. This makes that also because it's a gas and it doesn't deposit like closely. The problems of contamination were not in my region, but in Norway. And then this was one of the reasons when they started to develop the use of filters to avoid like these emissions. Before they were saying, yeah, we do just these very high chimneys and then we will not have here any problem. Not here, but 3,000-4,000 kilometers north in this case. Then the problem was to change the filters, that they were not changing the filters. So at some point, the filter didn't work anymore, but this is just more a political, economical decision.
Okay, thanks.
Okay, so thank you again. Wir sehen uns im Januar wieder. Schönen Abend und bis bald.